All right, title of my talk is Beyond Research, Clinical Utility for Cytokine Testing. Before I talk about how we test for cytokines, I want to give a little background on cytokines themselves. So as my mentor and long-term mentor, Harry Hill, used to always say, cytokines can be thought of as hormones of the immune system. So they're proteins that play an integral role in cell signaling. They're generally small in size, only about 5 to 20 kilodaltons. They can be involved in autocrine, so self-cell stimulation, if you will. Peregrine, so this is more cell-to-cell, so close interactions. And then also some endocrine fashion, so this is more through the circulation targeting other cells. So that's more what you think about hormones as well, where they made in one organ or gland and move through the circulation to their target. They're produced by a broad range of cells, including macrophages and B and T cells, mast cells, endothelial cells, and fibroblasts. They act through cell surface receptors and are important in modulating the balance between the humoral and cell-mediated immune system. So that's a lot of work we do. But they also play key roles in host immune responses to infection, inflammation, trauma, sepsis, cancer, and reproduction. So we got into the Luminex game quite a while ago. So I was reminiscing with Sherry last night, who we heard speak earlier. I think she started in 1999. I first visited Luminex, went down to Austin, Texas. I think it was about spring 2001. So we wanted to see what this newfangled technology was all about. We quickly saw the potential of it and started working with it. So back then they did have the Luminex 100 platform, but no XY plate handling abilities yet. So we're still feeding samples one by one into the Luminex instrument. So in 2002, we validated our first Luminex assay. This is a 6-plex panel for measuring cytokine responses to stimulated peripheral blood mononuclear cells. And I believe we're the first laboratory to ever actually offer testing based on this platform in a clinical setting. We followed this up a couple of years later in 2004 with a 10-plex panel for measuring cytokines in serum. A couple of years later, we then expanded this to 13 analytes, and we're testing both serum and plasma. So this is pretty much our existing panel now. It can be ordered as a panel, all 13. You can order it as a Th1, monokine, or Th2 panels, depending on which you want. And all the cytokines can also be ordered individually, with IL-6, IL-2R, and TNF-alpha being the most commonly ordered individual cytokines. So in 2020, we finally got around to converting it to magnetic beads. So this time we also wanted to perform a full revalidation of the assay based on CSL guidelines, and also establish reference intervals for serum and plasma and CSF fluid. Some of the other laboratory developed tests at ARUP include measurement of antibodies to the 23 serotypes in the pneumococcal vaccine. So we actually brought the 14-plex on soon after the cytokine assay. So this has been around since 2002 as well. Then we expanded it to measure 23 serotypes, so that's in 2 panels. We also have a multiplex panel for vaccine response to diphtheria, tetanus, and Haemophilus influenza B, and also for meningitis response. So this is, again, 4 different serogroups. So this brings us to the current validation. So what I want to bring to you today is kind of the perspective of a developer, so endpoint user. So what does it take to develop and validate an assay for a laboratory to develop test to start clinical patient testing? So we wanted some of the objectives of our revalidation, besides conversion to the magnetic beads, was to perform stringent and robust validation following CLSI guidelines. So currently there are no FDA cleared assays for cytokine testing. I think a few IL-6 assays received emergency use authorization from the FDA during the pandemic. So it's important then for both laboratory developed tests and research only tests to thoroughly validate these assays before we perform any patient testing or clinical trial studies. So we wanted to establish reference intervals. And again, I've kind of referenced the CSI guidelines that kind of apply to these different validation studies. Looked at accuracy, so this is determining correlation to our non-MAG and then the MAG-bead flex assay. And also looked at the NIB or WHO reference standards for the different cytokines. We did precision studies, typical there, linearity, detection capability, so determining limit of blank, limit of detection, limit of quantitation. Interferences, these were due to interfering antibodies, such as heterophile antibodies. And we found some real interesting results here, which I'll get into some more detail. And then also other known interferences in serum. This includes hemoglobin, bilirubin, triglycerides, and such. Looked at analyte stability, and as I mentioned, converted it to the magnetic beads. And this also really opened the door for automation of the whole assay too, so we were able to automate the assay using a TCAN fluent. I won't have time to go into any of those studies today though. So one thing that became quite apparent when I started development of the new assay were the rise of interfering antibodies. So when I initially developed the assay for serum, about one sample every few months we'd see potential heterophile interference that wasn't mitigated by our current specimen diluent. Before I started the MAG bead validation, I looked back at our pre-MAG bead assay and found that 4.3% of our results were labeled as unable to interpret due to presence of an interfering substance. So when this happens, we'll repeat the sample and then also do dilutions on that sample to try to figure out if there's heterophile interference there. So this resulted in almost 3,000 repeats, and most of these required medical director to review to try to figure out what was going on with that sample. So where are these increased interfering antibodies coming from? We don't know for sure, but speculation is there's more immunomodulating and cytokine inhibiting therapies now. So a lot of use with interferons, IL-2 in cancer treatment. The anti-TNF drugs have just skyrocketed the use of those in Crohn's disease and other inflammatory diseases. Then there's the IL-6 receptor drugs. And then we're also seeing a lot more immune dysregulation. So this is with CAR T-cell therapy, or as we just heard in the previous talk, we'll see cytokine storm with COVID patients. So heterophile interference, especially in these sandwich capture assays that we perform, which are highly dependent on rat and mouse monoclonal antibodies, they've been well known for quite a while. So back in the development of our original assay, we're quite aware of this. But back then we found that just adding, a little bit of FBS and mouse and rat serums to our specimen greatly mitigated the effects of these heterophile antibodies. So what are heterophile antibodies? It's kind of a generic term. They're just basically antibodies that interfere with our assays. So heterophile antibodies are endogenous proteins that bind animal antigens, as simple as that. They're produced against poorly defined antigens. They generally show weak avidity and they're multi-species specific. Both IgG and IgM antibodies have been identified, and they've been reported to be found in as many as 5 to 40% of normal patients. So these are healthy individuals. And as I mentioned, they're well-recognized cause of interference in immunoassays, as they can cause falsely elevated or suppressed results. Other types include your classic HAMA. So these are human anti-mouse antibodies that can result in treatment of patients. And unlike heterophiles, these interact strongly and specifically with the antibodies used in the assay. They can also be endogenous, naturally occurring human and autoantibodies, therapeutic antibodies, which I briefly touched on, and rheumatoid factor can also be classified as a heterophile. So how do these wreak havoc in our sandwich capture assays? So they can cause either a falsely elevated or suppressed result by bridging or blocking. So in the first example here, we have our Luminex bead with their monoclonal capture antibody bound to it. So let's call this for IL-2. Then we come back with our secondary antibody, which is again usually a mouse or a rat monoclonal against a different epitope of our target. So in this case, there's no analyte present, but the heterophile antibody can bridge those two antibodies together, so essentially causing a falsely elevated or false positive result. Conversely, here we have the heterophile antibody binding or blocking our capture antibody. So even in the presence of analyte, it can't bind and complete the sandwich, so you'll get a falsely negative or suppressed result. And the truth is that heterophile interference is pretty difficult to detect. Some of the ways we can do is we'll look at laboratory results, and if they're discrepant with the clinical presentation, this could be a clue. If the patient has a history of having received any anti-animal or therapeutic antibody preparations, this could be a clue. But we're a reference laboratory, so we test a lot of samples, but we don't have really any access to clinical information. So that's not very helpful for us. What we can do, though, is look for erroneous result patterns. So for the cytokine assay, for example, we'll look at the whole panel, and if 6 or more of the cytokines are exhibiting high results, so 100 picograms per mil or more, we suspect a heterophile antibody here. This is because in serum and plasma, normal patients, you really don't see a lot of cytokine. It's mostly undetectable. So to see that many elevated samples is suspicious. And then we can also look for nonlinear sample dilutions. So here are a couple examples I'm going to go through of some suspected heterophile interferences. So our first sample here, you can see 7 of the 13 results had elevated responses. These are all shown in red, so greater than 100 picograms per mil. So part of the workup, we will initially repeat that sample and then do a 1 to 10 and 1 to 100 dilution. So in this case, you can see that samples repeated well, but at the 1 to 10 dilution, where you expect concentrations, 10-fold less, we basically got all values were less than the detection capability of our assays, so less than 5 picograms per mil. So that looks suspicious. This next example really shows the havoc that these can wreak on our cytokine assays, these interfering antibodies. So 4 of the results looked normal. So a little bit of IL-10 response, 1 to 10 dilution, that put it below our detection limit. These 3 antibodies, or cytokines, showed, again, pretty good repeat results at the 1 to 10 dilution, so these results are multiplied by the dilution factor. You see an increase in values. So most of them are double or more. You go further on to the 100 dilution, and basically they disappear, so undetectable. Now looking at 6 others in our same panel. So here again, repeat well, 1 to 10 dilution increase, 1 to 100 dilution, they're even increasing more. So it's almost like the opposite of a hook effect, where you're seeing more as you dilute. So again, what do you do with these? It's really hard to interpret the results, and often we'll just say, looks like there's an interfering antibody. Results cannot be interpreted. Luckily for us developers, there are some more tools now available for reducing heterophile interference. So you can think of these as the conventional or passive blocking methods. So these include nonspecific substances like mouse IgG, animal serum, FBS, or nonspecific monoclonal antibodies. So this is basically what I was using in our first generation of the cytokine assay for heterophile interference. Again, these rely on binding of the human heterophile antibody to the blockers. And these are low affinity antibodies, so they don't bind well. We have these new generation of blockers now. Some of these are from Scantibody, and these contain specific blockers directed against the human heterophilic antibody. So it results in steric hindering, so those antibodies can then no longer bind to the antibodies in our assay. These are much higher affinity claimed, and they also claim to block all species, so mouse, rat, rabbit, goat, and as well as IgG subtypes. So for the first round of testing, I wanted to compare some of these heterophile blocking reagents to our current diluent and also look at some traditional mouse and rat samples or serums that can reduce heterophile interference. So a lot of data here. Again, multiplex testing. It's hard to condense it. So we're looking at our current diluent. So what I have here is a normal sample that I spiked with recombinant cytokines. So I just wanted some values for all 13 cytokines. So this is to see how these heterophile blocking reagents would interfere with normal responses. In a little more detail here, we're looking at IL-13. So in the current diluent, which was FBS and a little bit of mouse and rat serum, we're calling this 100%, so 192 picograms per mil. These are the 3 heterophile blocking formulations from Scantibodies. And again, good recovery, mostly close to 100%. The mouse IgG actually reduced our responses a bit. So this is just more visual. So the red dotted line is our current diluent, so we're calling that 100%. And you can see for our normal spike sample, we're getting good recoveries with the 3 heterophile blocking reagents. So they're not interfering with our assay. So that was good news. Next, we looked at a heterophile sample. So this one had 11 of the 13 cytokines were elevated. So looking at some of these in more detail. This is IL-13. You can see in our current diluent, very high values. Again, most patient samples are going to be undetectable for IL-13. When we put these antibody reagents in the diluent, you can see it pretty much totally wiped out these heterophile interferences. So hardly any values there. The mouse blocked about half of it. The rat serum really didn't have any effect on the heterophile antibody. Next, we're looking at IL-2 receptor. So again, current diluent, quite high values. Scantibody reagents reduce the signal quite a bit. So reduced it about 90%, especially compared to the mouse and rat serums. But for IL-2R, this is actually good news. So IL-2R is one of the few analytes in our panel where you do see a normal response in patients or in normal samples. So these 2 values, 6 and 700, are well within the normal range for IL-2R. So what this is saying is not only are these reagents blocking the heterophile interference, but they're also allowing us to quantitate the real or the true cytokine values in there. And again, kind of just more visual representation. So current assay, and then for all the different cytokines, so you can see the heterophile blocking reagents are reducing the interference quite a bit. Normal responses for IL-2 and 6 so far, and then it got cut off, but the others are the mouse and rat serums, which really didn't have much effect. So the last example is from a patient that was undergoing CAR T-cell therapy. So here we had elevated results for quite a few of the cytokines. But if you look at this pattern, so we have elevated IL-2R, IL-6, TNF, and then and 1-beta and IL-8. So these are real typical cytokines you'd see in an inflammatory response. So in inflammation. IL-10 here, interesting, that's more of a Th2 cytokine. So this is more down-regulatory. And we often see this. So the cytokine response, inflammatory response is ramping up, almost out of control here, or definitely out of control. And IL-10 is also increasing, trying to suppress this immune reaction. But again, what we saw here, so this is sample where these are true cytokine responses. They're going under severe inflammatory response. And again, the heterophile blocking reagents, we're able to see good recovery, so not much change in values. Mouse IgG I used here as well, and it was less effective. So a quick summary of these blocking studies. So these antibody reagents, we compared them to our current diluent. We found that they greatly reduced or eliminated heterophile interference, so I showed recoveries of about 90 to 100%. Assay controls spiked, and even our CAP proficiency samples all showed good recovery, so they weren't negatively affecting our assay. And this now allows us to actually report cytokine concentrations in samples with heterophile interference. So we no longer have to say the results are uninterpretable. Looking at some of the traditional, more passive blocking additions, such as the mouse and rat serums, they only reduce heterophile interference by about 20 to 80%, but they also reduce recovery by quite a bit, up to 40%. I promise I will get to some clinical utility. Another part of the revalidation, we wanted to increase the AMR of the assay. So the current assay, the non-mag bead, we looked at, I think for our AMR analytical measurement range was 5 to 1,000 picograms for our cytokines and 5 to 3,000 for IL-2R. We had a lot of requests from clients and also our clinical trial customers for better sensitivity, so less than 5 picograms per mil. We also had a pretty high repeat rate, looking back at the assays. So about 10.2% of our results were greater than the AMR. So we want to report out actual values. So we'd have to repeat these results at 1 to 10 and 1 to 100 dilution, so we could then back multiply and report an actual value. So increasing the AMR would reduce this as well. So I did this in a couple ways. Changed the standard curve dilution scheme. Actually cut it down from 9 points to 8 points. Put lower standard values in there, so 1.4 picograms for the cytokines, 4.1 for IL-2R. And I actually increased the dilution of the specimen, which if you think you're trying to get maximum sensitivity, you wouldn't want to dilute the sample further, but I think this really helped with matrix effects. So here's a summary of the data, LOB, LOD, LOQ. So current reference intervals were mostly less than 5, so in the 1 to 2 picogram per range in the low end, and then up to 4,000 to 12,000 for the cytokines and IL-2 receptor. Another thing we're real curious about is, we knew there are differences between the magplex and microplex beads. How is this going to affect the assay? How are they going to compare? So we looked at 60 serum and plasma samples, 10 spike samples, just for some extra values in there. Also our assay controls and our CAP, it's College of American Pathology. These are proficiency samples they send out to us for cytokine testing a couple times a year. Validation plan target was R squared values greater than 0.8, and we wanted slopes 1 plus or minus 0.35. So using Deming regression analysis, R squared values look fine, all greater than 0.9. We can see a few of the cytokines here, our slopes were higher than we wanted them to be. So what's this saying here is that overall, we're seeing higher results using the magplex assay compared to the microplex assay. So looking into these more, we pretty much attributed these differences to better recovery of our reformatted assay and the switch to the magnetic beads. So the magnetic beads allowed for more efficient washing. We're getting away from the filter plates, which always can be troublesome. The higher specimen dilution actually decreased matrix effects, so we're able to get better sensitivity. And then I think these HBR reagents also reduced interferences, so we're able to measure more accurately. Other evidence was that the mag bead assay showed higher recoveries for our known spike concentrations. These were samples spiked with recombinant cytokines. We also saw better correlation with our proficiency samples. So when I initially validated the assay, we looked at 40 matched serum and plasma samples. And this time we really didn't see a lot of difference between serum and plasma, so we just used the same reference range for both. We would test CSF, we'll test any fluid, but if it hasn't been validated or we don't have reference intervals, then report it with the disclaimer that it's an unvalidated sample type. So we wanted to validate CSF as well. So again, for reference intervals, we followed the CSL guidelines, which require at least 120 normal samples. So we had 130 matched serum and plasma specimens. So these were collected from ARUP donors. There were about 70 males, 60 females, and we tried to hit even numbers for each decade of life. So went from 18 to over 60. Inclusion criteria were basically they consider themselves healthy with no current illness. Exclusion criteria is any prescription medications. We did allow exception of birth control or hormone replacement therapy. And as I mentioned, we established them following guidelines and also using analyzed statistical software. And here's what we found. Definitely differences between serum and plasma. For the most part, we're going to see higher concentrations in plasma than serum. So again, a lot of these are lower undetectable IL-2 receptor. We get higher reference range. This also makes it kind of nice internal control in the assay. So if you don't see any IL-2 receptor response in there, then you can suspect maybe the incorrect dilution or misdilution of the patient sample. For CSF, what was interesting is we actually saw quite a bit of IL-8 in the CSF, so much higher than both serum and plasma. Also looked at analyte stability, just quickly here. So the conclusions were cytokines don't mind being frozen, so 30 days frozen, and even 3 freeze-thaw cycles, there's less than 20% change from baseline. Ambient storage, forget it. Even after 2 days, 1 day, the cytokines will rapidly degrade. Refrigerated, for the most part, 1 to 2 days of storage was fine for most of the cytokines. TNF, though, is very sensitive, so very labile, so it degraded rapidly even overnight at refrigerated temperatures. So if you can't test the samples right away, these are all received as critical frozens, so we'll thaw them and test them. If we need to repeat anything, then it's better to refreeze them than to even keep them refrigerated, especially for TNF alpha. So a summary of our validation. So we incorporated these heterophile blocking reagents in the specimen diluent, and as I showed, they greatly reduced heterophile interference, and our repeat testing and non-reportable results went down quite a bit. We were able to increase the AMR 4-fold. This also reduced a lot of repeat testing for samples that were greater than the analytical measurement range. We had better recovery with the non-mag bead, or compared to the non-mag bead assay, and better agreement. And then we established independent reference intervals for serum plasma, and we also validated CSF as a sample type and established reference intervals for it. So as promised, we'll get into a little more clinical utility now. So when we first brought these assays, cytokine assays online, they thought we were crazy. So this is a research tool. Again, this is 20 years ago. It's like, who's gonna order it in a clinical setting? But we went forward anyway. And if you, in our laboratory test directory under utility of the assay, so this is, if you look up our serum cytokine or our cytokine panel, it says right there, primarily for research to support attempts to understand the pathogenesis of immune, infectious, allergic, and inflammatory disorders. But since we brought this online, volume has just steadily increased. And currently there's no really established diagnostic utility for cytokine testing that relates to any particular disease or illness. Of course, when I said this, my medical director came up with an exception, and that was IL-2R is used in the diagnostic criteria for HLH, which is hemophagocytic lymphohistiocytosis. But it's a receptor, not really a cytokine, so I stand by my statement. Cytokine testing is gaining greater clinical utility for monitoring cytokines and immunomodulating therapies. So we see interferon alpha and beta used to treat hepatitis and multiple sclerosis, IL-2 in cancer treatment, and then interferon gamma to treat CGD. And as I mentioned, these anti-TNF drugs, infliximab, adulimab, and there's several others now. Humira, you probably see commercials for them all the time on TV. And IL-6. So not only for monitoring these different therapies, they're also used, we heard a little bit about safety monitoring, so looking for cytokine storm and other things in clinical trials. And then also monitoring cytokine storm in CAR T-cell therapy. And then always we're involved in studies as looking at these as potential biomarkers for inflammatory disorders in either peripheral blood or the central nervous system. So some clinical utility, still a lot of research applications. So this is looking a bit at our cytokine testing volumes. There's going to be a quiz here. So looking back through November, again, we lost some. So the black line is IL-6 plus our panel testing. Pink line was IL-6 only, and then we can see, or the black is combined, and then it got lost, but that was for the panel testing. So you can see pretty steady volumes here. This is like 2 to 3 3,000 samples a month that we're testing for serum cytokines. And then about March, April, we just saw this huge increase. So about a 30-fold increase in cytokine testing. A lot of that was IL-6. So does anyone remember what happened? April 2020. You're laughing and smiling. What was it? It was our pandemic. So this is when it first hit U.S. shores. This was when the pandemic, cytokine storm. So this was the pandemic when it hit New York. So our cytokine testing, especially IL-6, just skyrocketed. Then later you can see there's another spike here. So this was correlated to the Orlando outbreak. That's before they started shutting everything down. This was interesting. So besides IL-6, these clients from Florida were also ordering TNF, IL-8, IFN gamma, IL-2, and IL-10. Again, if you remember back to the slide I presented on the CAR T-cell therapy, those are all the same cytokines that were elevated in that cytokine storm. So why IL-6? Why were doctors so hot in using IL-6 and testing for it? So you got to remember, this is early days in the pandemic, so we had emergency use authorization to validate our molecular assays, but these are still being validated in the labs and hadn't been offered yet in large volume for patient sample testing. So the physicians were using these to predict cytokine storm syndrome, and then using them to guide therapy, so either administering steroids and or IL-6, and then monitoring the patient afterwards. So they're ordering IL-6 on their severe COVID patients every 24 to 48 hours. So the idea was here, you wanted to not hit them with steroids too early and suppress their immune system so they couldn't do a natural response to an infection. But if you waited too late till full-blown cytokine storm, it's going to be too late and they weren't going to be of any benefit. So a little more on cytokine storm. These were patients who demonstrated excessive immune activation. This immune dysregulation is characterized by perpetual activation of the lymphocytes and the macrophages, so just releasing large amounts of cytokines. And these would lead to overwhelming systemic inflammation, multiple organ failure, and high mortality. So this was a study by Luke et al. So they looked at IL-6 concentration. So this was from 2020, so right during the pandemic. And so they looked at 24 patients that, for ICU admissions, so that were under the respiratory distress. And what they found basically was that IL-6 levels had a 1,000-fold increase. This was compared to their normal control group. When they looked at IL-1 beta and TNF, they saw about a 2 to 10-fold increase, but nothing as much as IL-6. Other studies also showed that IL-6 was better than CRP, ferritin, liver enzymes, and other clinical laboratory markers for predicting outcomes of respiratory failure and death. And they actually established some reference intervals or some cutoff points here. So I think this last slide I'm going to present, for the most part, is really illustrates the versatility and adaptability of the Luminex platform. So what I didn't mention is I told you I was revalidating the assay for magplex usage. But right in the middle of the validation is when the cytokine storm hit and our cytokine testing skyrocketed. So for IL-6 testing, we'd run the full panel and then just report out IL-6. So what happened here, we were burning through kits, running the full panel and reporting IL-6. We're going through kits faster than we can make them. So what we're able to do then is quickly spin off IL-6 only assay. So this is looking at correlation not only of IL-6 in the panel, but this was the microplex versus the mag bead. So again, between the two, we saw good correlation. Then linearity studies looked fine. So within a couple weeks, we're able to quickly spin off from our 13-plex panel, a single analyte panel for IL-6. So as you imagine, this reduced costs quite a bit, and it's a lot easier to develop a single analyte panel where you're only coupling one bead and having to use one conjugate than trying to couple and QC 13 different cytokines on 13 different beads. So in summary, cytokine testing is now routinely performed in clinical reference laboratories. As I mentioned, there's no FDA-cleared in vitro diagnostic tests approved for cytokine testing, so both laboratory tests and RU assays should there be thoroughly validated. And I showed us examples how to do this following CLSI guidelines. There's pretty much guidelines for all of these. I showed some clinical utility. So IL-2R does have some clinical diagnostic utility. It's also used for monitoring cytokine storm and immunomodulating therapies. We see a lot of use in safety monitoring for clinical trial studies, and then some examples of cytokine storm for both CAR T-cell therapy and COVID-19. And so as a scientist and test developer at ARUP, we specialize in more esoteric tests. So it's my job to continually look at new methodologies, new technologies. And it's been 20 years now, pretty much looked at every other multiplexing platform out there. And I think Luminex still remains a really robust and versatile and flexible platform that we've stuck with this whole time. I mean, as a laboratory test developer, we really like the flexibility of it. We're able to do all the coupling in-house, control that. We don't have to send anything out. There's no really specialized instruments you need for spotting. So yeah, it still remained a very good and reliable assay for us. And did I get us back on time for lunch? Most importantly, any questions?